Hello, hello.
Hello, sir.
How we doing?
And how are you?
Good, good, good. Good, good, good.
We'll send them by email after the event. This webinar will be recorded and it will be available on our website, and you will also receive an email with a link to the recording, yourselves and anyone else that has registered. Now I'll hand it over to Dr. Gomes.
Thank you very much for the introduction, Maura. Again, I'm Connor Gomes, the lead developer on the SCISSOR instrument, and I'll be talking today about how we're developing an assay to simulate the prodrug biotransformation in vitro and conserving those enzymes that cause that biotransformation within our SCISSOR platform for use with prodrug injectates. Really quickly, once the slide changes. Just to give a brief overview of what we're going to be covering today. I think most people won't need a whole review on the drug development process that are in this webinar, but I'll just briefly touch on the points that I want to highlight before continuing on with our conversation to get into more injectables and then the prodrug environment and the unique problems that are found using traditional dissolution methods to analyze prodrugs. And through that, we'll then introduce the SCISSOR platform, how we're able to overcome those barriers, and then we'll get into the actual use case of the system in a case study that we went through at Pion. So really quickly, from the start of drug development to the end, there are many different people involved, different instruments, and different methods of analysis that are all trying to probe different characteristics of the final drug form or modality being created. Down in drug discovery, we have the single identification of APIs where you're looking for your drugs of interest. And then the preclinical research, which will be the bulk of our conversation today, will involve the in vitro testing and in vivo animal testing that is needed for characterization of the formulation itself. And then before going into the clinic and then drug approval via your governmental bodies. Pion focuses directly on this in vitro testing, and we develop a whole bunch of different in vitro testing platforms or platforms that are able to aid in the analysis of these drugs during preclinical research stages. They all have to do with some sort of a solubilization process and tracking some sort of solubilization of a drug of interest over time. I won't get into them now. We'll review some of them later and how they're apparent and relevant to the study that we're going to be talking about today. But specifically, I'm going to be talking about those that deal with injectable drug modalities. So I have here a brief schematic of showing just using skin and where we normally inject as an example, nothing really deep, at least nothing really deep in this slide. We have your intramuscular injections. You can think of your COVID vaccine if you'd like, where you have a pretty sizable needle and getting into the 10 mil range of administration, where you're injecting deep under the skin into a muscular collagen-filled, a lot of solid and aqueous components found there. And the diffusion of the drugs that are injected there will depend on their interaction with those solid architectures, as well as the chemistries that are happening in the aqueous environment. And there are a few cells there, mainly fibroblasts, that will have the primary interaction with the drugs deposited. So moving up a little closer to the skin, we have our subcutaneous tissue. This is where the conversation will steer today, as the platform we are talking about is the subcutaneous injection site simulator. In the subQ space, there's a wide range of needles and deliverability methods in order to get your bolus situated within there. You can see the wide range of volumes going up to multiple tens of mils, given correct circumstances. And a lot of the same processes that dictate solubility and diffusion that are found in the intramuscular space will also dictate that in the subcutaneous. The only difference is there's going to be a much greater ratio of your interstitial fluid and a little less blood circulation and less solid material as compared to the muscle. Moving up a little bit, we have intravenous. We're all very familiar. I think most people would be familiar with this mode of drug delivery, where you have a very small range of needle sizes and you can administer as great a volume as the human body will take very quickly. And then lastly, just a small mention for intradermal injections, normally with very thin needles because you're going in between your layers of skin, very small volume because that's all that can fit there. and it's a pretty slow uptake that's basically dictated by passive diffusion. Except there are a lot of tiny, really small vesicles that run through there that will eventually uptake your drug. So within these spaces, there's a whole bunch of different ratios of the components that are responsible for drug uptake, drug diffusion, and drug metabolism. And so normally you have some sort of aqueous buffering agent, typically around neutral because we're human beings, but that will normally be a bicarbonate buffer found in your interstitial fluid, your blood, etc. There'll be a range of ECM and proteins that all these drugs will interact with, and they will be different based on where you administer these APIs. So for example, in the muscle, you're going to have a very large concentration of collagen there. And with younger neonatal sort of drugs, you're going to have more fibronectin and elastin present instead of those collagen residues in the same tissues. There's going to be a lot of glycosaminoglycans. These are long chain sugars between a kilodalton and a megadalton in size. The reason these are primarily important, even though they're lay biomolecules soluble in that aqueous environment, they're large enough where they can actually sterically and architecturally wrap and interact with your drugs, which then impact not only passive diffusion, but it also affects the rate at which maybe an enzyme of interest that needs to act on that drug, it can now not see it because of the presence of these glycosaminoglycans. The cells, although a very important part of drug metabolism, it won't be a large part of what we here at Pion talk about. The cellular processes that happen internally and externally from a cell can really only be replicated by those cells themselves. So the adipocytes in the sub-Q and the fibroblasts in the intermuscular, not really present in too many in vitro tests, especially ones I'll mention today, but they still do the job that we need them to do to process the API. And then lastly, you have transport mechanisms that get your either excipients or drugs from where they're administered to where they need to be, whether that's in a small local environment in and outside of a cell, or they're completely transported to a different tissue in the body. All of these components are going to affect, and the concentration of these are going to affect the rate at which your drug is absorbed, which is sort of the measurement that we're all looking to get out of an in vitro assay when we're talking about preclinical development in the formulation space. However, when we look at, when we try to extrapolate and hypothesize how our drugs of interest will interact in these spaces, there's a few in silico methods and other methods we can discuss later if anyone has questions. But there are these molecular, micro, and macro structures that will always be apparent that aren't going to really be captured by your traditional dissolution methods or your stirring beaker dissolution methods that we're doing on a benchtop. So, for example, in the subcutaneous space, although you do have a large interstitial space that's going to be filled with a bicarbonate buffering system, interstitial fluid, glycosaminoglycans, there are going to be different stages of interaction, as in when you first administer the drug, the large part of that interaction is going to be those things solubilized in solution because your bolus hasn't really set in and interacted with all the solid structures yet. However, when your boluses do ended up interacting with the solid structures, there's going to be a lot of different ratios of proteins and enzymes doing different metabolic actions on the molecules of interest. In the sub-Q, this is an example of just small details that can matter a lot. We have a surface of an adipocyte, which is the type of cell that we normally think dominates the subcutaneous space. So what that means is that we really wouldn't have a large concentration of collagenous residues. But as it turns out, on the outside of those adipocytes, you still get collagen matrices that are aggregated on that surface. So even if you're probing a drug that is supposed to go into the sub-Q and you're not really thinking about collagen, you are going to have, not overly significant, but you are going to have interactions every single time you inject into that space that may not be captured with a simpler system. Moving to the intermuscular, it gives a really good sense of the microstructures that can be found there and where bolus and solid crystals and particles can actually aggregate within human tissue. I think the debanding on collagen fibrils shows that really well, where in a nanometer range, you get these small deviations in collagen fibril structure, which will then impact the surface area interactions that your bolus and APIs will have within that environment. And again, it's just these small details that normally aren't, that sometimes aren't captured in the hypothetical scenarios that we're trying to built in our brains when we're trying to guess and extrapolate how these drugs are going to act in the clinic when all we're working with is aqueous buffers. Even more so than that, when we look at the macrostructure, when we're developing types of in vitro assays in order to probe these types of interactions, we kind of have to think about whether we're injecting or administering into a diseased or a healthy state. I have examples of heart tissue here where pre-stroke you have nice aligned collagen fibrils. What that will cause is a higher rate of diffusion through that space when you administer a bolus. So you're going to have channels where the flow of least resistance will exist, and that is where your bolus and your API will be leaching from. But if you look at the disease state, that is not the case. When the wound healing process is happening, collagen is dumped sort of in that space, and typically there's a layer of fibronectin that helps align everything. But as we get older, that fibronectin goes away, and as we get injured, that process breaks down, and this is what you're left over in a lot of your tissues, especially after injury, which after injury, when you're getting medical intervention, is also the space in which you would maybe administered a subcutaneous injected or intramuscular injected drug. So taking all those factors into account and wrapping it back to in vitro testing and how we actually go through thinking about how to in vitro test on these drugs, there are a couple methods I mentioned, but I'll review them quickly on what we normally jump to. So the very end goal of these preclinical studies and the most widely used during the drug development process when it comes to getting that the governmental body data that you need is animal studies. So animals have very similar, not equal, but similar metabolic processes, similar tissue geometries and architectures and buffering capacities and chemistries that are happening within their biological tissues. So it's a great way to see how your drug will react in a whole body model for pharmacokinetic and toxicology assessment, which are normally replaced by a multitude of in vitro tests. Now the actual most common one, because it's also the cheapest, would be the USP2 dissolution apparatuses, where it's a very standardized method of doing a beaker stirring dissolution method, tracking drug dissolution in aqueous media or otherwise. Very cheap, very standard, and will be part of every drug development process. And then moving up in complexity a little bit, you have your USP4 dissolution apparatuses, where you have either poorly soluble or long-acting drug formats or modalities, where you have passive flow over that material of interest, and then you track the small amount of solubilization that's happening over time. I highlighted a couple of the pros and cons, but again, animal studies are fantastic, but they do fail to accurately predict human subcutaneous outcomes. This has been outlined in the literature, largely because when we're moving up in complexity of drugs, when you increase the surface area of interactions, when you have a large modality like a mAb or a protein, due to just the bulk interactions that happen on the surface of those molecules, all of the other chemical interactions that happen within those tissue spaces are going to end up mattering more and more and more. So as we've made more complex drugs, we've also gotten worse at having our animal models correctly predict the clinical outcomes that are actually going to happen for a human. USP2 is a fantastic method, except it doesn't replicate the subcutaneous environment. And we'll get to this with prodrugs, but it's very difficult to include any metabolic enzymes or metabolic processes within that apparatus. And then the USP4 improves on USP2 because of the more modalities in which you can test within the USP4 apparatus. But again, it doesn't emulate the subcutaneous injection and only simulates in bulk fluid, which again causes problems when you're trying to integrate those native metabolic enzymes and proteins that are responsible for things like biotransformation of prodrugs. So although there are all these shortcomings, surprise us, oh, sorry about that. We have one more thing to point out, is that from a regulatory standpoint, all of the tests I just outlined are very important and have been used in most modern drug applications. And they're all needed, the in vitro and animal tests, so you do get a good idea of your pharmacokinetic, metabolic, toxic, and clinical performance before you actually inject into a human. However, there's not a lot of guidance when it comes to subcutaneous injectables. And not only that, there's the recent FDA push to minimize the amount of animal testing, not only for the ethical and financial reasons, but also from a scientific standpoint of actually accuracy of predicting those clinical outcomes. So here at Pion, we have our SCISSOR N3 platform, the subcutaneous injection site simulator, which we try to develop to overcome some of the barriers that I just listed. So the SCISSOR will incorporate many of the physicochemical proteins constituents that I mentioned previously, because we are able to hold in a reaction cell an artificial extracellular matrix where you will deposit your bolus of interest. And then that bolus of interest will diffuse within that biorelevant environment to an outer acceptor chamber, just like many of the other dissolution apparatuses that we all are familiar with. And then the drug concentration is characterized there to then be related to systematic uptake within a human. This, again, as outlined here, it's a quantitative measurement of drug release, which can then be related to a clinical value. But also there's other analytics involved within the instrument where you can get qualitative measurements of things like bolus shape, bolus diffusion, turbidity, the rate of API crash out, and a whole bunch of other things. So within the instrument, I'll be largely focused on this cartridge at the heart of it, because that's what we'll be talking about. And that's where the artificial extracellular matrix that I previously mentioned will be located throughout these assays. This artificial extracellular matrix, you can see it visualized here in the camera, will have one pH probe at the top of it that will monitor the pH at the injection site. You have a pH probe in your acceptor chamber, which will hold a bicarbonate buffer. As we already mentioned, that bicarbonate buffer is very important for accurately predicting how a subcutaneous injected pharmaceutical will act within the body, because that's the buffering system that it is first that is shocked with when it is administered. We can control the temp via thermocouple to track at 34 degrees Celsius is a normal assay, as that is the temperature of the subcutaneous tissue in a human. To inject, we typically use a 1 ml pre-filled syringe format or something very analogous, which will administer the drug at the top of this cartridge near that pH electrode, near the injection site. And then to actually monitor the amount of API or really any constituent that is leaving this artificial extracellular matrix into the outer chamber, we normally employ an in situ fiber optic probe with our Rainbow platform, where instead of pumping fluid anywhere, we instead take absorbance measurements at the end of this, fiber optic probe, and that is how we can get our in situ and immediate data as to not worry about how much transformation of these drugs, APIs, excipients, and other constituents is happening when it's incubating in aqueous environment at pH 7.4. But without the Rainbow, there's ability to offline sample for offline analysis via something like HPLC. So how are we going to use this assay and these analytical methods to analyze prodrugs? So a prodrug, just to review, is a compound of an API of interest. This API may be insoluble or maybe only have to be active within a certain tissue within the body or a certain subset of tissue within the body. And attached to that API, either to solve its insolubility problem or its specific mode of action problem, a promoiety or some sort of R group is attached to make your entire prodrug, the square and triangle. The prodrug will most likely be much more easily formulable for the formulation scientist. It will be normally soluble within an aqueous environment, so that way it can be administered easy. However, the API, when it is attached to this promoiety, will be inert. It will not be able to be active until that promoiety is cleaved. So that promoiety is normally cleaved by a native biomolecule. This biomolecule can be found immediately at an injection site before circulation, so that it was just easy to deliver the drug, and then it can become insoluble thereafter, and it can still go through its normal mode of action. This biomolecule can also be present in a specific tissue away from the area of administration. So you can think of a cancer tumor, for example, has a few specific enzymes that can act on a specific few R groups. And so if you want say a chemotherapy, something that you don't want active in the whole body, only active within that tumor, you can tune this promoiety to only be cleaved by a biomolecule found within that tumor. Therefore, you only have your active chemotherapy or other cancerotoxic, I'm sorry, that's the incorrect word, drug only active within the cancer tissue. So The promoiety that's cleaved off will normally be biosafe and can be excreted via normal human methods. And then you have cleaved off, you have your active drug, which is maybe insoluble or soluble, but in this example, it is turning now insoluble to then therefore be active. What this means is that within the SCISSOR platform or any dissolution apparatus where you are probing the action of prodrugs or the uptake of a prodrug or the cleavage of a promoiety from a prodrug, you're going to have the enzyme of action within that assay. I give an example of esterase on the screen here. Esterase is the most common enzyme for all of prodrugs that have been cleared by the FDA to act on. I believe it's over half of them. So I visualized here. This esterase will then act on these prodrugs to then cleave at an ester to then release your active drug. This esterase, this will become important in a moment, has a 10 angstrom tunnel in which these molecules have to funnel through in order to be cleaved by the esterase with a small 4 angstrom cofactor or pocket, which is shown here. We looked to integrate esterase into the system. So the first, in order to start to probe these prodrugs and see if we can actually track not only the cleavage of the promoiety, but the systematic uptake of the cleaved drug. So using paraben esters, we employ esterase to cleave off, in this example, just tracking the activity, a methyl from a methylparaben molecule to then result in 4-hydroxybenzoic acid. And so all we did was in a vessel incubate our methylparaben with the esterase. And I've shown the examples of the HPLC chromatograms here, where at about 4 minutes you see the peak for methylparaben, and then at about 2 to 3 minutes you see the 4-hydroxybenzoic acid peak. And so just by tracking the growth or disappearance of these peaks, we're able to probe the activity of our esterase molecule. And so this over time, we are able to track and qualify the activity of the enzyme. The establishment of this assay meant that we were able to qualify the enzyme's activity in all stages of the SCISSOR ECM manufacturing and implementation within an assay. So what we did was we took our artificial ECMs, which again is where you are administering the bolus within the SCISSOR platform. So we took all those artificial ECMs, we integrated the esterase into our manufacturing lines, and at every manufacturing step, we were able to sample off some of that artificial extracellular matrix and do this activity assay to make sure that no constituent we were adding into our artificial ECMs were affecting this enzyme's mechanism of action, and therefore won't affect any assay we have on any prodrug that's injected. And just here I outline a pretty reasonable conservation of enzymatic activity within that ECM through all steps of manufacturing, storage, and then an actual 20-hour assay. So I've just listed the activities here for that validation work. You can see that in a buffer, we get a much higher activity. And that's because within our artificial ECM, we have things like the native glycosaminoglycans and other constituents that can be found within the sub-Q space. And so the viscosity and the pore size and the relative permeability of that material is going to drop as compared to a buffer. So by about 1,000 times, we drop the activity of the esterase, but it does conserve this activity throughout the duration of a SCISSOR assay. And we have data that's not shown within this webinar, but we have a little bit of validating data where enzymatic activity can be conserved after 6 days of manufacturing these ECMs, which opens up the door for longer acting assays. So after validating that the esterase and these enzymes can stay active within our system, we looked to expand upon our learning set outside of methylparaben to see if we can actually emulate the kind of action that you're looking for in an in vitro test within a formulation pipeline. So all we did in order to probe that was we increased the chain length found off of these esters, off of these parabens. And the molecule we're looking for, again, is 4-hydroxybenzoic acid. and these paraben esters, methylparaben, butylparaben, and octoparaben, will all be cleaved off. Those carbon chains will be cleaved off. And hopefully we get to see release of multiple species. I want to point out that octoparaben has this 8-carbon chain, excuse me, which will mean it will largely be insoluble in aqueous media, and that's what we saw on benchtop. So notably, if there's mainly any release of octoparaben going forward, we know that our enzyme's active in it, and it may be a good molecule to see actually how much activity you can get when you are simply doing a strict insoluble to soluble transition. So in order to track the release of these drugs, we employed that rainbow system I spoke of with the in situ fiber optic probe within our acceptor chamber. The 4-hydroxybenzoic acid, you can see the, the solvent used to solubilize all these molecules was NMP, N-methyl-2-pyridine, pyrrolidone, and that peak, the peak of the NMP is this one at 232. So all the absorbance after that is coming from the esters and carbon chains that are added on. And so with 4-hydroxybenzoic acid, you can see that we have this peak right before 250. And then with a little bit of more conjugation or ionizability from the carbon chain, you get a peak shift closer to 300. So the distinct peaks are initial evidence that we should be able to distinguish between these two molecules in solution using our fiber optic probes. But if the direct spectra does not work, we also have the ability to transform that data into a second derivative spectra. And within that second derivative spectra, we have a shift of this peak at 250, a little higher, that we may also be able to track as a unique signature. So the first drug that we're going to mention is the methylparaben injection. So this is not the activity assay of the esterase, but an actual injection within the SCISSOR platform. You can see the images at during the injection, then 1 hour and 9 hours into the experiment, where the injection looks, you can clearly see the NMP with a different dielectric as compared to the aqueous buffer, but then pretty quickly that dissolutes, and then you're left with a transparent matrix, because the methylparaben will be soluble within aqueous solution. So again, what we're looking for in the direct spectra, just as a qualitative measure, is this peak at 250 and this peak right before 300. And you can see that there's a little bit of both in the spectra, which is to be expected, as again I mentioned, the methylparaben is going to be soluble as well as the 4-hydroxybenzoic acid within this assay. So although both are going to be released, both are going to be coming out. So the ratio between the two will strictly be dictated by the amount of action that the enzyme has on the methylparaben. And just to sort of further prove the point that the negative second derivative peak that we need in between 250 and 256, it's about right in between them, which is anecdotal evidence that we're probably getting a mixture of these molecules released. But moving on to the other spectroscopic signatures, let's move on to butylparaben with a longer carbon chain. So the butylparaben molecule, you'll see, has much less of a significant release right below 300, but we still get a pretty significant shoulder right below 250. What that means is there's probably a slight mixture, but butylparaben should be a little less, have a little less aqueous solubility than the methylparaben. But after transformation in the 4-hydroxybenzoic acid, we should see solubility of the paraben or the ring, and then we should be able to detect that via UV vis. And just in the second derivative, it's just a little messy, but it sort of just shows the negativity of that peak. So we are getting release of the species, but it's kind of hard to deconvolute within this space. And then lastly, the injection that I feel is the most interesting is the octoparaben, because this octoparaben should not really be soluble at all and should only be released after action of the esterase. And you'll see noted by that red line, there's almost no signature around 300 with the octoparaben, but we do get that shoulder starting to show up at 250, which is again qualitative, at least gut check, that we do have the enzyme acting on our injectates. And so, but as you can see from all the spectra I just showed, they're not the prettiest. They're like, we're tracking shoulders and we're tracking, like qualitative shifts that I'm explaining verbally. In order to deconvolute those release curves, we employed some tools we have within our AU Pro software system, which is the software package that's paired with our Rainbow platform. And there's just a multi-component analysis deconvolution tool, where here we have the red, blue, and green standards, which are our 4-hydroxybenzoic acid, our methylparaben, and our NMP spectra, respectively. And you'll see their predicted spectra is the dotted line. I'm sorry if it's a little small, but the predicted spectra using these tools on the statistical side, it's able to build our spectral signal using certain weights of the 3 colors. And then we are able to get 3 release curves of our 4-hydroxybenzoic acid, our NMP, and then the uncleaved paraben molecule. So after employing this method to all of our inject dates and going through sample by sample, I just want to show the release of the cleaved species. So we have on the right, you can see the graph of the cleaved methylparaben, the cleaved butylparaben, and the cleaved octylparaben by that multi-component analysis. And this is the result. So as predicted, the rank order is correlated to the molecular weight of the promoiety or R group that we have attached to the drug, where methylparaben not only has the highest aqueous solubility, but it also will have the fastest cleaving action. The butylparaben, next on line, will have much lesser aqueous solubility, but after cleaving will have similar and would be released slower naturally. And then the real one of interest is this octylparaben, which had the lowest release and a pretty long lag time, which means it took it looks like it took the enzyme almost an hour for it to start to act on that molecule, where you can see that there's almost immediate liftoff of the methylparaben peak. And just to show this, we have the octylparaben spectra shown on the right up here, where compared to the NMP, where the octylparaben control experiment, where we're injecting that molecule into a normal standard ECM without the presence of esterase, we get almost no release, as compared to the butyl and the methyl, which still have a little bit of release within that standard ECM. The octylparaben really highlights the enzymatic action. Note that these are normalized releases because our control experiments, because of the presence of all those carbon chains in solution, is really affected our analytics. And so further down the road, when we end up cleaning this data and put out a white paper, that this control experiment will actually be directly comparable to our prodrug, so that we can actually see how much that enzymatic activity is lifting up this drug uptake. So that about sums it up. In conclusion, we were able to validate enzymatic activity, specifically esterase, which acts on over 50% of the FDA-approved prodrugs. We were able to have that enzymatic activity conserved for 1 to 6 days within the platform. Not only that, we were able to show an insoluble to soluble transition with kind of all of them, but specifically octylparaben, to show that the biotransformation of that molecule into its soluble form may be modeled within SCISSOR. And then on top of that, our Rainbow system and AU Pro software are incredibly helpful in deconvoluting the samples, so we can track these promoieties, solvents, and prodrugs, as well as the cleaved drug, within one complex matrix. So thank you for your attention. Thank you for dealing with a couple of my small rants in there. If you have any questions at all, there were any questions asked during the presentation, I will gladly answer them. And I just want to thank you for your attention. I have 2 contact informations on the screen. You can see mine up there, where you can email me for any question, anything you have at all. And I've also put up the product manager of the SCISSOR platform, Imi and SSC's email up here. If you have any more like sales-based questions, can probably go to her.
Thank you so much, Connor. We do have a few questions.
Lovely.
The first one is, what is the thickness for each skin layer?
Oh, wow. Off the top of my head. So I don't recall the exact thicknesses off the top of my head. In my, yeah, I'm not 100% sure on each thickness.
Yeah, I think Imi knows. Imi, do you want to comment?
Yeah, sorry, I just some relative thicknesses probably around for the epidermis, maybe 0.05 to 1.6 millimeters. And then we're probably looking at the dermal layer around 0.3 to 4 millimeters. And then the hypodermis obviously can fluctuate quite a lot depending on the individual. But we're normally looking anywhere above 1 to 2 millimeters. But again, that can differ based on the individual. Sorry for cutting in, Connor. Go ahead.
Thank you, Emmy. Thank you. Another question, Connor, how long does enzyme stay active under the testing condition?
So under the testing conditions within the assay, we have up to a day of that release, or at least confirmed stable release for 12 hours. We have in an assay, we have validated enzymatic activity within that cartridge up to 20 hours. And so we have in storage conditions up to 6. So the only left to validate is just running an assay where we're able to conserve the material in there for 6 days, which is a factor. It's a more complicated factor, but and then just doing the same exact activity assay thereafter. So if it is a question of interest, Pion does have the ability to really see how long these enzymes will stay active, and if we have the ability to refresh that activity.
Great, thank you. A few more. Are there any limitations on the molecules that can be integrated into the Scissors artificial ECM?
Yeah, the standard ECM and the ECM presented here is aqueous based. So it would be great if we weren't trying to include some sort of highly lipophilic molecule. And the only real limitations are going to be on the activity side. So I think the integration of any protein is we have the know-how to do a lot of them. It will be increasingly tricky when you work towards molecules like fibronectin and collagen, where the way you integrate them into aqueous solutions and hydrogel-like materials can actually dictate their structure and function. So there's really no limit, but there's definitely things to consider when you're integrating certain biomolecules.
Thank you. Another question is, esterase is a group of enzymes, probably with different selectivities.
Mm-hmm.
Can your assay be used on other ester prodrugs?
Yes, we can integrate specific esters into these artificial ECMs. The esterase you see here is generally purified. It's a general esterase purified from bovine liver. But if there is a specific tissue and a specific enzyme, that is not a barrier for Pion or for the SCISSOR instrument, frankly. So as long as the material can be sourced, it can be integrated within the artificial ECM.
Great. And then do we need to run a control with samples for each test? How do we prove the system is working properly?
Yes. Again, with something like octoparaben and insoluble to soluble transition, it is pretty easy to tell, because if you expect enzymatic action and you have enzymatic action on that API in a benchtop like beaker-like experiment or a 96 well plate-like experiment, you should also get that action set scissor. So the control experiment would be good because it would compare your solubility of your prodrug to the scissor solubility of your cleaved prodrug. Oh, sorry, my brain got away from me. Oh, control experiments. So you will need to do control experiments simply because you would want to run the same drug without the enzyme present. But as far as validating the enzymatic activity with these experiments, you shouldn't need to do that every time. We found that, add in similar mass of enzyme, you get similar activity.
Okay, thank you. There's a follow-up question on the esterase. Are cartridges with the esterase commercially available yet? And if so, can you specify what type of esterase was used in this application?
Mm-hmm. Yep. So the esterase in this application was if you need something more specific, please send me an email. But it is esterase from bovine liver. I'm pretty sure on that. The best case scenario would obviously be human esterase. There's a sourcing and financial difference to them, but that's not a barrier. Amy can comment more on our ability to send these out or have these integrated in a customer-facing fashion. Typically, these are employed on site. So we have analytical services where we run internal studies. That's where this type of ECM is right now. And I obviously can't expand on it, but we do already have one validating study where we did run, excuse me, a formulated prodrug that was being developed.
Okay, thank you. Next question. The artificial extracellular matrix simulates the subcutaneous tissue, which can be considered a complex structure present in the cartridge, while the outer chamber represents the interstitial fluid that exists between the cells in the subcu tissue. Is this correct?
That is a reasonably good generalization. Really the acceptor chamber is meant to be that sink uptake. So whether that be lymphatic, circulatory, what have you, that is supposed to represent the passage through that permeable membrane. Within the ECM system itself, it is supposed to represent the subcutaneous space in bulk, as it is a complex space. This is an acellular assay. There are issues with batch-to-batch reproducibility with cells, which is why on your site it's a different conversation. But at least from Pion's standpoint, it's a very hard thing for us to do. But yeah, the systematic uptake would be equivalent to that lymphatic or circulatory uptake. And then as it's sitting in that chamber, it is supposed to simulate as it's sitting in between those adipocytes in that interstitial fluid. So,
thank you. Okay, a few more. Do you know, is there a ready-to-use cocktail available to simulate the ECM for sub-Q administration?
Yeah, we have our standard ECM here at Pion. It's what is our typical consumable ECM for our scissor assays for a very wide range of drug modalities and companies and targets. That is our ready to go mixture, where you receive that in a pre-filled syringe, already aqueous, already ready to go to put right inside the scissor for your assay.
Yeah.
Outside of that, Pion is always open to having more development-based or research-based conversations and anecdotes and anecdotal research avenues. So if there's anything more complex that you're asking about, we are absolutely open to talking about either one, how to manufacture it batch to batch here, or how to figure out a way to reproducibly get you making the same thing over and over and over again.
Thank you. A few more. Great webinar, Connor. Lots of great questions. Is there any way to do fluorescent imaging of the scissor cells to highlight or tag certain components?
We have integrated a 720p visual spectrum camera. We do not have the ability to take fluorescent images. I promise you that complex image analysis is on our development dinner plate, or on the stove. I'll say it's a better analogy. So we have that going. You may sample off. So as I mentioned, one, fluorophores sometimes can be picked up in UV-vis region from secondary spectroscopic processes. So it is possible that if you do have a fluorophore, that fluorescent tag can still be detected via UV-vis. I'll just say that one anecdotally. But no, we do not have integrated in the scissor or rainbow a way for you to do a fluorescent-based imaging assay. That would have to be done offline via auto-sampling from HPLC.
Okay. What was the membrane size of the cartridge? And was the esterase only confined within the cartridge and not diffused into the larger chamber?
Yes. So in this experiment, the esterase could diffuse into the larger chamber. We have a 5-micron pore size on that membrane. And the artificial ECM, the glycosaminoglycans found there, and the proteins, enzymes that we integrate, that's a pretty big pore size. It will diffuse through, and it's sort of meant to, because you have material turnover within the sub-Q. If you didn't get that material turnover, you would have very little passive diffusion and then release of your drug thereafter. And so, however, you do point out a great thing, that you will still have enzymatic action in that bulk solution. So if you do have, say, a prodrug that hasn't been cleaved, release, it will then be cleaved thereafter, and then you'll get the signal. So on something like HPLC, it will look very different than something on Rainbow. That being said, we have the ability to manufacture the inner cartridges with various pore sizes and membrane materials. So we do have membranes that we can drop down that pore size so the enzyme does not leave, has a very small chance to leave, so that all enzymatic action can happen within the cartridge. So in this experiment, things were free flowing with a massive bias for them being in the cartridge, but there are ways in order to, I'll say, fix to be a cleaner experiment where you would slow everything down, but you wouldn't get diffusion of that enzyme out of the cartridge.
Great, thank you. Is your assay sensitive enough or for a high prodrug amount will be needed? Have you performed any assays with long-chain fatty acid prodrug molecules?
I may have to defer to Amy here if she remembers if we have any long-chain fatty acids and if she remembers the concentration that we were able to detect.
Yeah, and if we can't answer it here, we can always follow up with the email.
Yeah, for this one, we may have to follow up offline. I think off the top of my head, I don't have that sort of information, but we can definitely look into our archives and double check for you.
Great, thank you so much. Is this something that we can purchase from Pion, and what would it look like to work to conduct a study like this?
Yeah, so that would depend on what your real want is from Pion. There's a different conversation that happens if you have a scissor platform in-house or whether we're doing the work for you. Pion has relationships where we can do bulk assay during drug, standard development pipeline things, CRO-like action, where we can do everything of the drugs that you send us. Or if you have a scissor platform, I don't want to claim anything. There's a large range of mechanisms where we can get the molecules you want inside the system, whether it be from us or sharing information appropriately for the company on your site to actually start doing all the chemistry yourself.
Excellent, thank you. Next question. It appears the local estrus concentration decreased in the cartridge over time. Would there be a strategy to keep the local estrus concentration stable within the cartridge, or how could I verify the concentration change wouldn't affect the cleavage efficiency over time?
Yes, great question. What I mentioned with the membrane, the pore sizes would be the most straightforward and most inexpensive way to actually achieve this. There are more that is what I would recommend. What it does is you lower the pore size, so the diffusivity decreases. So you slow everything. You have more of the chemical interactions happening within the cartridge, and so by the time you get release, all those interactions happen more as a chromatographic method by the time it reaches that membrane before release. So that's like what my immediate answer would be. On top of that, just on a material science standpoint, I'm going to guess, I don't have the method off the top of my head, but if you needed something to stay in, there are many hydrogel, polymer, and biomolecule based manufacturing methods in where you can tie the enzyme you want to a solid structure. So we don't have that at Pion, but that could be an answer to your question.
Excellent. Well, thank you so much. That's about all the time we have for today. I want to thank you to Dr. Connor Gomes for your contributions and your wonderful presentation. And thank you to our participants for being a great audience that made this interactive website possible. Be sure to check our website for upcoming webinar and events. Thanks again for joining us, and we'll see you next time.
Thank you, everyone. Have a good one.